Welcome to Don't Fear the Wait podcast with your host Vicky Masita, bringing together science and personal experience.
Hi guys, welcome to the next episode of Don't Fear the Wait with your tiny titan host, Vicky Masita. That is me, although I was known as the mighty midget last week. I don't know whether that's a compliment or not, whether we should keep the tiny titan or whether we should kind of move over to the mighty midget. I'm not really sure. But anyway, enough about me. I have got an extraordinary guest on today by the name of Andy Galpin. Andy, how are you this morning in bright, sunny California?
I am fabulous this morning.
Oh, amazing. I love it when people say that they're fabulous because it actually means that they're telling the truth.
Yeah.
It's so boring when people kind of go, yeah, I'm good. How are you? But the fact that you're saying you're fabulous.
Hate it. I'm doing amazing.
Right? Life is too short to be boring, Andy. That's what it's all about.
You betcha.
So anyway, I'm going to hand the mic over to you. So tell us a little bit about you, your background, what you're doing over there in California for the people who obviously don't know you.
Sure. So I am an associate professor at Cal State Fullerton. So I am the director of what's called the Center for Sport Performance. And within that, we've got 7 different laboratories. And one of those laboratories also happens to be mine, which is the biochemistry and exercise physiology or molecular exercise physiology lab. So really my job is 3 things. We study what makes muscle grow, shrink, die, repair, but generally in the athlete end of the spectrum. So we don't deal much with disease treatment, prevention, management stuff. We deal with elite athletes. So we try to study all things muscle, and we take muscle biopsies. So we look at this from the cellular level all the way down to the molecular and genes. And but we try to answer practical questions and training questions and performance questions with that. So we're not sort of using exercise as an intervention to study biochemistry. We're actually trying to study performance. So really that's for the most part what I do. And with that, then I work with athletes on small occasion, usually pretty elite ones and pretty selectively, but that's really what I do, study muscle.
Right, fantastic. Now, forgive me if I'm wrong, but I'm basically going to kind of harp on about your research that you're doing because I am fascinated by it. And some of the research that you're delving into at the moment is muscle fiber typing. Is that right?
Yeah, I've probably done, there's probably just a few people in the world that have done more than me at this point. So yeah, we can say delve into that. That's fair.
Let's delve into that then. So now again, I'm not a big science boffin. I am part of the Team Science group or the hashtag, should I say, on Instagram, hashtag Team Science. But let's have a look at these muscle fiber typing. So you're looking into research about what the best kind of exercise techniques are depending on the muscle fiber types that an individual is born with. Is that right?
Yeah, I mean, we do really everything fiber type. We try to understand why people have certain fiber types, what you can do to change them in particular directions. And I mean, we've seen now everything from, of course, we've known for a very, very long time that your training does it. So the type of training you do causes very specific changes in the fiber type. But even more recent things have shown that phytochemicals, polyphenols, macronutrient splits, carbon dioxide concentrations, and then even recently a paper came out showing that cold water immersion, all of these things change fiber type. So I think we can maybe to start the conversation, people understand that, yes, of course, you're born with a certain fiber type percentage, so a certain number of fast and slow at the very rudimentary level. But outside of that, it is extremely plastic. Everything you do matters, and they change in response to just about everything. So that's really what we focus on.
Right. Okay. So let's take it then from a muscle hypertrophy type of standpoint then. So for bodybuilders, if they wanted to progress in the best way possible, would it be recommendable for them to find out how many different types of fibers that they actually have, and then how they can train to augment it, I suppose?
Not yet.
Okay.
That's the honest answer. And I really, really wish I could say a different thing. But the honest answer is not yet. That is the hope. And I will say that is absolutely what I will probably say in the next few years, if we can really have the ability to continue our research. But there's almost no question. We have some fundamental basic background stuff that suggests that's exactly what's going to happen. We just don't know the answer yet. And that's the really hard time I have giving you a thing, because no one wants to fund this type of research. No one cares about these things. You're probably aware, like in the States, it's just very little funding for muscle research if it's not obesity or things like that. So at this moment, no, but that is probably very likely to be true in the near future.
Okay, that's amazing. So is there a way then that you can maybe give us an idea on how to increase power then? Maybe power or endurance based on fiber typing?
Yeah, so I mean, there's a lot of things. Maybe we can back up for a second. Let me add more to that hypertrophy question.
Okay.
So we know we have a good indication. Classic dogma tells us fast twitch fibers are bigger and more likely to hypertrophy. Slow twitch would be the opposite. And in fact, even our research from about 10 years ago now showed very clear molecular differences between the two fiber types. So it's not just a activation issue. I hear this one all the time. And that's complete garbage. We have hundreds of papers now showing that just the molecular environment within a fast twitch and a slow twitch fiber, it's different. And the molecular environment such that it activates muscle protein synthesis or growth or hypertrophy is just much more concentrated in the fast twitch fibers. That's innate to the fiber. It has nothing to do with activation at this level. Activation totally matters, but there is something intrinsic to the fiber types themselves that suggests fast twitch fibers are probably more likely to hypertrophy. In addition, we have some speculative research suggesting that if you do, say, your classic hypertrophy range, 5 to 8 to 12 at the most repetitions per set, that's going to selectively hypertrophy the fast twitch fibers. And no one's really been able to confirm that because people either don't have access to the single muscle fibers or the imaging techniques, but we do. And so this is actually something that Brad Schoenfeld, who I'm sure some of you are aware with at home there, we've been trying to collaborate in this for a long time, and it's just we just can't get. We've actually been trying for years to get funding for it, and we can't. But we could really answer this question very, very quickly if we could just come up with some resources, because it's not that difficult. With the technology we have some pretty amazing things in our lab that no one else in the world has, and we have access to an almost $1 million microscope, and we can do some pretty cool stuff. So I think, going back to your original question, Vicky, that if we can identify that, say, like with Brad's research suggesting, hey, maybe sets of 20, 25 repetitions up to 30 does equal whole muscle hypertrophy, it's very possible that that is actually activating slow twitch fiber type specific hypertrophy. And the lower ranges, your 5 to 8 to 10 to 12 are doing fast twitch. Now that's all speculative at this point, but if we could identify that, then we could say, okay, get some testing done. You'll know what your fiber sizes look like. And that way, so for example, if your fast twitch fibers are 5, 10 times the size of your slow twitch, maybe they're getting pretty close to their ceiling in terms of the maximum size they can get. So maybe at this point, you should turn around and target the slow twitch fibers to make the whole muscle itself grow a little more evenly. Again, though, that's why I had to answer your question, though, saying the honest answer is no. But I could guess, I could speculate. I actually had this conversation with Ben Pekulski recently.
I remember.
And he was saying, he's like, look, I feel like if I would have done more stuff and targeted my fast twitch fibers better, I would have actually had more gains. And so that's the problem is, okay, if you go and identify that your slow twitch fibers are lagging behind, I don't know if that means you should make the slow twitch better, or if you should say, hell, slow twitch and go the other direction. And that's why I can't give you a very specific answer, but we're getting a lot closer to these concepts. And I think that ultimately will be the thing that allows us to be much more effective in our individualization and personalization of training prescriptions. We're just not there yet.
Okay, that's fair enough. So for argument's sake, realistically then, obviously, if we can't go and punch holes in people's quads and things like that to get the biopsies from them, and you're saying we can have overall good muscle belly development, would it not be clever enough to do a periodized training program then to kind of target the 12 to 15, 20, 30 rep range, and then in the next step, go to like your 5 to 8s?
Well, okay, I think you've actually stumbled upon, and I say that jokingly because I know you are not guessing there. You're very smart. But that would be the thing I would say for right now. What the practical takeaway from what we know now is to say, okay, everyone should maybe run that whole spectrum of hypertrophy ranges. And if you give it, say, conservatively 3 to 5 weeks, kind of at a certain window, you will probably notice something taking off or not. And that'll give you a lot of insight of saying, hey, look, maybe I've always been at this 8 to 12 range, and now all of a sudden I did some sets of 30. And if you look at like what Louis Simmons and Westside people did, they went up to sets of 100 sometimes.
Yeah,
and like, I think it's really good to go, you know what, it's been, I remember doing this years ago and being like, man, it's been at that point years since I've done anything over 15 reps. Like, okay, you're probably insufficient in those areas. So you go and do a set of 25, 30, and all of a sudden you're like, okay, like something may grow where it hadn't been growing for years. So I think that is the practical takeaway right now is you should explore a little bit over that entire rep range, even go outside those numbers. I mean, Brad showed up to 30 works, but who cares? Do 35, 40. There's no magic here, right? You don't know what's going to not work, and experiment from there. And then you might identify. And I had this happen with a student of mine, actually, is he kind of did that and went years. And then all of a sudden was having problems, was looking back to his training logs and was like, man, I don't understand what's going on. Looked back and realized, like, years ago I did these sets of 35, went back to him 2 weeks later, boom, took off.
Wow.
Started growing again. So I think that is a very important thing to do is let's open up our minds a little bit and train across a wider range of spectrum. But where this gets even more confusing is this is all assuming that the repetitions are equated.
Sure.
So if you want to get even more controversial here, or I'd say more confusing, we could start talking about, well, how isometrics count or eccentrics count, or
drop sets, rest pauses, intensive training. Sure.
Because that throws everything into completely out of whack. So, but the concept, anyways.
That's it. So it's interesting because a lot of people as well look at power builders. So they actually do maybe 2 compound movements between the 4 to 8 rep range, and then the remainder of their training session will go 12 to 15, and then they'll do a couple of kind of what we like to know from John Meadows, the ape shit sets. So they'll do like 30, 40, 50 reps and drop sets and rest pauses and use all of these intensity techniques. Now, I suppose in a way, where do you go from that if that's where you're starting from? Because it's not like, even if you run a 4 to 6 week bout of that, you can't realistically just kind of go, okay, I've been doing that now, let's just go straight onto 30 rep sets, because you're already doing that. So it's not a new stimulus, right?
Yeah, but I think you can, in terms of, you could turn the whole thing into 30 rep sets. You could you could change into isometrics. You could change into slower movement, or the opposite. I think where depending on kind of which group, how you get into bodybuilding, some people also forget that you can go fast with your repetitions. It's okay to explode and contract really, really hard. And that is oftentimes, again, depending on kind of which camp you came in from, oftentimes a forgotten aspect. So I would say maybe go the other end of the spectrum. Or do some other things, because like if you're like, so for example, if you're doing that where you're hitting a little bit of almost like a vertical integration approach where you're hitting a little bit of everything in the same day, same week, well then maybe you just take a whole day and you just do one thing and you just emphasize the 20s or 30s, or you emphasize the 5s and you don't go over 5s, things like that. So there still is a lot of variation you can have, and you can change up the approach. And I think that, as we look at more and more research and looking at trying to identify the molecular mechanisms that induces muscle hypertrophy, I think one of the things that's completely clear is that the muscle doesn't know a lot of those things. It doesn't understand at that level. It just knows sort of how much fatigue, how much stimulus is there. I think within that range, you can really do a lot. And so I think what that tells me is that you should explore a lot of variation. Don't confuse variation with randomization. Like that's not the same thing. The difference there is planning. Right? At least that's how I teach those words, right? Variation is randomization with a plan, right? Randomization is doing random weird shit.
Yeah.
That's not what we're after, but you do that in a structured, organized fashion. And then what you'll start to identify, and this is actually some of the things that Ben Fikulski preaches, that, hey, like that stuff is kind of noise. It's really about focusing on the quality of the contraction. I know Brad just is about to kick off a study where he's looking at posing. So you do a set and you pose in between, and then you go back and do it, whether or not that actually changes muscle hypertrophy. And some other work has shown that like the muscle-mind connection. So thinking about the muscle more, trying to contract it more. And that's actually now, again, this is what bodybuilders have been saying for decades, if people would listen. And now the research is going, actually, maybe there's merit to that too. So I guess why I'm saying this, Vicky, is to say, like, some of that stuff we don't have to obsess over too much. Let's really spend more time emphasizing the quality of the exercise, the quality of the repetition. That probably matters a lot more than if you're doing a drop set or 21s or pyramids or whatever you're doing, are you taking it to the same reasonable fatigue? Are you getting a full contraction? Are you contracting the right part of the muscle? And are you really emphasizing quality? Can you feel the difference? And, of course, I'm biased here. This is what my book was about, but really it's, can you feel what's going on? And if you can do that, the rest of it is just a stimulus that you're using to help you get to a certain internal feeling. And all that is there, and it just helps you kind of organize things in your mind, but that should be the ultimate goal.
Excellent. Yeah, I really like that. I follow a really good bodybuilder. He's coming on not so long in time, called Seth Feroci, and he is a really, really big advocate of posing in between his sets. Mm-hmm. And it's funny because he always goes on saying, well, I don't give a shit what people think about me when I'm posing my biceps in the mirror, when I'm training biceps, because that way I get a pump, my bicep is full of blood, and et cetera, et cetera. So it's true, they do have some merit to it. So we all harp on about the bros in the gym who do a rep and then pose, but actually it may be very viable for them. So that's excellent. Okay, so let's delve into some of the external factors that can manipulate your muscle fiber types and actually obviously pose some kind of impact onto them. You were talking about cold water immersion. Now that's interesting, because I have a few people at the moment who are actually advocating having cold showers first thing in the morning. Mm-hmm. Is there what's the idea behind that? So
those are really sort of separate ideas.
Okay.
Something like a dosing of cold shower in the morning would really play no role in muscle fiber type change. It wouldn't be probably enough of a dose. And I should back up too, to be forthright. The data that just came out on that it's very speculative at this point. It showed just that the genes that induce muscle fiber type changes were activated in response to cold. But this is pretty cold for like you're talking cold shower or cold bath for a couple of times a week. And it didn't actually show a fiber type change. They didn't get that far. They just showed the genes that induce it or activated. So it is very speculative at this point. But I would be sure that if they continue to do the next trial where they actually measure the fiber type, they will see a change. So, but having said that, the shower thing is really other issues. It really wouldn't induce a fiber type change at the level of a cold morning. A cold shower in the morning. It's not that cold. It's more sensory cold than it is physiologically cold.
Right. Okay. That's fair enough then. So what about other external factors then? Things like you were talking about, maybe chemicals or nutrition. Can that influence things?
Yeah. I mean, there's been several studies. High fat, high sugar diets can do it.
Oh, wow.
Oh yeah, for sure. That's a very common one. You can look at things like resveratrol. The people are aware of that. I've seen some studies done with apple concentrates that have changed fiber type as well. So everything you do matters. Resveratrol has a very specific molecular pattern that we're aware of, and so we actually know the chemistry and the biochemistry behind that. And so it's not only like, oh, that's speculative. Like, no, we know the molecular mechanisms it's directly doing. Like, we know the mechanisms behind it. It's not fake. So everything matters. People used to think, for whatever reason, well, I actually know exactly why, but we want to get into it here, that fiber type can change. And we actually have submitted right now for peer review a study that we did in twins, and this is monozygous twins with the exact same DNA. One of them had stopped exercising when he was about 18 years old, and the other one had never stopped, and they were both in their early 50s. So we had about 30 years of two people with the exact same DNA, one of them exercising. Now, this wasn't hypertrophy training or anything. He was an endurance runner and a triathlon.
Okay.
But it's exercise nonetheless, right?
Sure.
And the non-exerciser was about 50-50 fast twitch, slow twitch. And the endurance runner was about 90% slow twitch.
Oh, wow. Okay. Big, huge difference.
Yeah. And so really, I mean, we've seen this in as little as 11 days in space flight fiber type change. We've seen this in a lot of those as 4 probably weeks or so in normal people just detraining. So it happens both directions, fast to slow, slow to fast. It happens extremely fast. And it really, really, really matters. So it's, in fact, the honest way, because I get the follow-up question is always, well, how much can you change? And the answer really is indefinitely.
Okay.
It only comes down to exposure. And exposure comes in load or intensity and time. And so given enough time, there's no reason you'll stop to change. It's just simply, did you run out of time, or did you? So the more you expose it to something, and the more time you give it, the more change you will see. And I mean, we've seen this actually with some pretty elite level bodybuilders slash powerlifters, that their muscle fiber size is literally off the charts. We're about to release one very, very, very soon of a person that you would be very, very, you would know exactly who this person is, and his fiber size was, I'll put it this way, that the size, we had to start looking at other species because we couldn't find another human that had an equivalent fiber size.
That's incredible. So he's actually a mutant.
Yeah. So like my point is, everything is plastic. There's, your genome is set, like you can't change the DNA of the genes you inherit, but how that stuff is expressed is so much more reliant upon your lifestyle than we've ever understood. And the better we get with technology, whenever we get an improvement in technology, when we go back and measure something that we thought doesn't change or doesn't work, we almost always find out, oh, it turns out it does. And it's almost always an issue of technology. So that's the one thing I would caution people on, is you have more control over these things than you think. It just, for some of us, you may need more time or exposure. Some of us need less, depending on the genetic starting point. But we all have not necessarily the same ceiling. So we all can't get to the same place, but we all have the same relative range, right? We can all move a tremendous amount within our own spectrum. And that's what I think is important for people to understand, is that spectrum is a lot bigger than you think.
Definitely. Really interesting as well. Now, just as a, because I've just kind of like gone off on a tangent in my brain as well of what to ask you on that one. Obviously, these muscle fibers that you're going to be releasing on this well-known bodybuilder or athlete, I would say, is much larger than anything else that you've ever seen before. Does that actually include muscle fiber hyperplasia as well? Or is that literally just or can you double up on the fibers, or what is that in general?
So the way that if your bicep gets bigger, you have basically one of two options. Number one, the actual fibers within the muscle get larger themselves, so they increase in their diameter, or you increase the number of fibers, or I guess third would be a combination of those things. Now, we'll kind of glance over this part, but in case people are wondering, there sometimes is the idea that there's certain types of training you can do that increase sarcolemma specific versus, like, that's actually never been shown at all. There's no evidence for that silliness. So I won't say it's not true because it would be hypocritical, but there's no evidence to suggest there's a certain type of the cell that gets bigger and the other size doesn't get hydrated or gets swollen or something like that. Like that's all noise. Having said that, the classic dogma is that people don't hyperplasia under normal circumstances, but I think actually if you look at the evidence, it suggests quite the opposite. The term that you want to look up is fiber splitting.
Okay.
That's the term, that's the scientific term, because we don't really hyperplasia in the common sense where you grow a new fiber out of nothing. What happens is a fiber itself kind of splits in half, and they're very small and they each hypertrophy. And we have extensive evidence from the animal world that happens. And if anything, humans are much more plastic and more adaptable than any other mammal for the most part. So it would be really weird for us to think we don't do that. And we have, again, we have some other imaging stuff from our lab that suggests it's probably happening. So one of the things that's difficult is, in order to really understand that, we have to be able to somehow count total number of fibers in a leg or an arm or a shoulder. And that's really, 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 really difficult. And so we use indirect measures, like we take the volume of the whole muscle with an MRI or something, and then take the volume of the individual fiber size, and then you can, of course, just divide, right? It's not that simple, but basically, right? And what we do see for sure a lot of times in bodybuilders is they have a much higher number of total fibers. So I can't say that we'd have to have a biopsy of them when they were kids before they train to see if the fiber number goes up, but the indirect evidence, the ones that have done things like that, do suggest that fiber splitting is happening. It just probably takes a lot of time. Right. It probably takes a lot of eccentric work as well. And it takes time. It's not going to happen after 6 weeks or 6 months probably, but I would probably bet my career that after a few years of training, so people like, me, I don't know when you started training, but I was probably 13 or something like that.
Oh yeah, I was a bit older than that.
Okay. But like, I trained for 20 years. I've trained for 20 years. Like I know for sure there's some fiber splitting that's happened there. And so I don't know. We'll have to see. We're gonna have to get some more bodybuilders in the lab and get some more high level muscle people in terms of muscle size in the lab. But yeah, that's one thing I'm very, very confident of, that it's not only it's possible, it's not a 50-50 split between hypertrophy and hyperplasia. Yeah. But I think we have to stop saying it's impossible or it doesn't happen. It just may happen at a lower scale than traditional hypertrophy.
Amazing. Now, one thing I'm really interested in, you might not be able to answer this question, and I bet you won't, because it's the story of my life. But
well, I don't hesitate to say I don't know on podcasts. So like you just start
no, that's brilliant, and I love it. Have you been able to do these studies on women as well? And have you seen any difference?
So, general answer is no.
I know.
Actually, in about, I don't know, 3.5 hours, my graduate student Nathan will be defending his thesis, and we actually biopsied 16 elite female weightlifters, and these are world caliber, gold medal holding these kind of athletes, and this is Olympic weightlifting,
okay,
specifically. And we haven't got a chance to analyze these data for fiber size yet, but we did analyze them for fiber type. And the traditional dogma is that women have more slow twitch fibers, right, than men. And I can give you the sneak peek here, but you can't tell anybody.
Don't tell anybody yet. Got
it. That's not the case. We found them to have very high percentages of fast twitch fibers, even as high, if not higher, than the men.
Right.
If anything, we've seen before. It's as far as I know, it's the highest percentage of fast twitch fibers ever biopsied in females, and the reason is, to the best of our knowledge, there's only been about 4 studies that have ever been done on fiber type of female strength or speed or power or anything anaerobic athletes. And of those 4, you're talking studies that had 6 girls in it or 4 girls in it. I mean, it was something, we calculated the numbers, but it was something insane, like 35 or 39 females have ever been biopsied for their fiber type, female athletes.
Sure.
Like total, not studies, like total women. I mean, we do that in every single guy study. It's just like
exactly right, and it's infuriating. It is infuriating because women do still want to know the science behind stuff about them, not just from what it pertains to men.
Right. So we had that. So we'll be coming out with that stuff soon. And then actually this summer, my postdoc Irene Tobias she's a PhD in biochemistry, but she's also a power lifter, and she's actually mixes that up with triathlons. Just for fun.
Just for fun.
But she's been all about this for years now. And so we've finally done enough of our due diligence. And this summer we'll be starting a very impressive trial where we're going to be looking at all females, and we're going to be looking at fiber type specific molecular responses to high intensity exercise.
Amazing.
So it's going to be really, really cool. We've been gearing up. We honestly wanted to start about 8 months ago, but we couldn't get any funding for it because they still won't pay funding for females.
I know, and it really is. It's so annoying. Well, if you need another athlete, I'm quite happy to come to California for you. And I can take part in a study in California. That's fine. I can do that.
Come on out.
I will. Cool. Right. So I think that leaves us on a really good note, Andy, because I know that obviously you are time constrained as well, and you're going to start teaching. So I really do thank you for your time, because I know that you're insanely busy. But just before you go, can you just tell the listeners where they can find out a little bit more about you, whether they can donate to your funding, and where can they follow you for your research coming up?
Yeah, sure. So social media is easy. Twitter and Instagram at Dr Andy Galpin. It's pretty easy to find. You can go to my website, andygalpin.com, and that's actually a website where I don't sell anything. I don't have anything to buy or anything like that. But what I try to do is take all my university lectures and all of my conference lectures, and every time I give a talk, and I put it up on that website, and I just give it away for free. There's no newsletter to sign up for. There's no membership pro or anything. Just give all that stuff away. And I'm a little bit behind right now, but I've got a whole couple of semesters worth to catch up on and to upload. So hopefully I'll do that time. And there's a Patreon account attached to that if you want to kick in $1 or $2. But if you're interested in the science, you can always email me about contributing. And to be honest with you, 99% of the history of my entire lab has come from donation.
Amazing.
And you can do that from our website. It is, we are actually a charity by American definition, so we can give you a tax receipt if that matters. People have done a lot of that and kick back on their taxes and things. But it goes directly to our lab. I don't take salary out of it. I've never paid myself $1 salary from that fund. We use it straight for research or for our students to present things like that. So that is how our lab survives. And doing biochemistry and molecular research of hypertrophy is very expensive. So we appreciate every $5 or $10 or more, or especially the bigger ones. If anyone's got $500 or 5,000 or 50,000, looking for a place to hide in some taxes, just give old Andy a call. I know what to do with it.
That's it. Brilliant. And then what I'll do is I'll put in all of those links underneath the YouTube video, guys. So if you do want to obviously contact Andy and put your hand deep into your pockets to help fund this research, because let's face it, we all want to know the best way how to get big quick, right?
Yeah.
So,
and my email's pretty easy to find, so you can link directly to my email. I don't care.
Fantastic.
It's just agalpin@fullerton.edu. So.
Brilliant. I'll get all of those links over to you. Andy, thank you so much for your time. I really appreciate it. And guys, that's it from Don't Fear the Wait this week. Look out for some new guests as well coming up. We've got a busy podcast lineup this week. So lots of love, and I will speak to you all soon. Ciao.